everyone. Thank you for the invitation, and it's my great pleasure to share our animal work on GRIN variants. Okay. Part of study in this talk supported by Seed Therapeutics. Our team at Emory started this human GRIN variants project in January 2012. In the past near 14 years, we developed the ability to obtain data describing multiple functional parameters of the variants. We integrated these parameters for quantitative evaluation to define the variants functional standards. There are so far near 1,000 disease-related GRIN variants. We are trying our best to work on it as many as possible. We believe this in vitro functional analysis is helpful for the clinical stratification, development of mechanistic hypothesis, and also on selection of the variants for making animals. So to further evaluation whether the variants underlying patients' conditions, we developed a full knocking model at Emory. We also are working with collaborators on another 6. I will focus on this E4-3G. If I have time, I will introduce some recently published SA10 data. For the GRIN mice, we do general evaluation, electrophysiology, spinoanalysis, immunostaining, immunoblood, MRI expression, PET scan, and also a set of behavior tests for motor coordination, locomotion, learning memory, anxiety, social interaction, sensory motor gating, and the seizure threshold. For the therapeutic strategies, we evaluate FDA drugs, co-agonists, PAMs, NAMs, newly developed compound, and several genetic rescues, which are in the different stage. This E4-3G variant was from a female with a profound developmental delay. She rode by 6 months, sat by 19 months, walked by 4 years, and spoke only single word mom at 5 years. The mutant residue is located in agonist binding domain. This residue does not contact with the agonist directly, but it interacts with other residues to stabilize the sealings of the binding pocket. Molecular dynamic simulation predict that these variants create a tunnel allowing water to access to the binding pocket and makes the agonist more accessible to water, which may disrupt the stability of the binding pocket and facilitate the agonist on binding. Indeed, this variant significantly reduced the glutamate potency by 54, which means activation of this receptor requires much higher concentration of agonist. PET lab data on Hexel show that E4-3G has much smaller counter response. If we normalize these traces, we can see the response time was much shortened. Also, E4-3G significantly decreased the surface expression. We integrate all these parameters, taking together 3 high confidence functional reduction, including reduced glutamate potency, shortened the deactivation toll, reduced surface expression, along with less than 0.1% of this charge transfer. Therefore, this is a strong loss of function variance. The heterozygous knocking mice host these variants, reduce the body weight with a smaller litter size. We first evaluated the effect of the variants on synaptic activity. We evoked the EPSC from CA1 pyramidal cells by stimulating cell collateral. We can see the E4-3G has a much shortened decay time and much smaller current amplitude. We also record miniature or spontaneous EPSC. E4-3G saw a smaller MDA current amplitude, also shortened decay time. We then investigate the influence of E4-3G on LTP. We apply this theta burst stimulation on cell collateral and record extracellular field potential in CA1. We measure this slope of the field potential normalized to baseline and plotted here, we can see clearly E4-3G decreased the enhancement of the field LTP. This data indicate that E4-3G significantly altered the synaptic activity. It is well known that spike dendrite plays an important role in linear memory to evaluate the influence of the E4-3G on the dendrite. We cross E4-3G mice with Taiwan YFP mice. The image of YFP expressing C1 pyramidal cells were imaged, reconstructed, and analyzed. We can see the E4-3G significantly reduced the spine head diameters with reduced the mature mushroom type and elevated the immature thin spike type. It has been established that the spike structure can be served as a proxy for the levels of synaptic activity. Therefore, we think this spike change may reflect the accelerated EPSC and diminish the field LTP I showed before. We then collaborate with Dr. Catherine Roche's lab at NADS to evaluate the influence of E4-3G on the MDA subunit expression. This immunoblot showed that E4-3G has significantly reduced GRIN2B protein level in this total homogenate, along with an unexpected elevation of GLU-A2 signal. GLU-A2 is a AMPA receptor subunit. However, in the PSD, synaptic fraction only showed GRIN2B reduction. We then evaluate the influence of the E4-3G on whole brain activity by dynamic in vivo PET scan. 3 PET markers were evaluated: GRIN2B ligand, SV2A ligand for synaptic density, and the last one is FDG for the glucose metabolism. We can see clearly E4-3G has significant reduction in the GRIN2B ligand uptake as well as SV2A ligand uptake, but no change in FDG uptake. This data suggests that E4-3G significantly reduced the to-be-mediated excitation as well as the synaptic density, but has no effect on the cerebral glucose metabolism. The next question is, do the E4-3G mice show any behavior phenotype? We did a locomotor activity test for consecutive 2 days. We can see for the wild type, the exploration on day 2 is much less than day 1. Well, for the E4-3G, the exploration between day 1 and day 2 is quite similar. This data indicate that the E4-3G mice may not remember the same box they entered in on day 1 and re-explore, may reflecting potential memory deficit. However, the water maze test show a comparable performance except for a faster swim speed. Field conditioning, so more freezing, which point the mice to have a better memory. But it is well known that both water maze and field conditioning are kind of stressful, maybe confound the test, especially for the mice with altered anxiety. I will show the data next slides. We therefore choose this bar maze test, which is less stressful, which requires animal to explore, to locate, and enter into escape chamber, put it underneath one of the 20 holes. We can see in the first 4 training days, the E4-3G mice require longer time to find the hole with the escape chamber and enter into the chamber. On the 5th day for the probe test, the escape chamber was removed. Then the E4-3G mice took longer time to find, to search that probe hole. Well, and also they spend less time around that whole area. This data suggests that E4-3G display an impaired spatial linear memory. Yeah, we then the elevated zero maze, which is widely used for anxiety study for rodents. The animals preserve less access, more willing to explore bright, elevated, and open arm, opposed to staying in the enclosed and darkened arms. This wild-type mice wild-type mouse above, not afraid of this setup, and crossed open arm many times, while this E4-3G mouse below stay in one side of the enclosed maze almost all the time. This data suggests that E4 and 3G display high anxiety level behavior. And we then did pre-pulse inhibition test. Here, you can clearly see the E4 and 3G mice has reduced percentage of the pre-pulse inhibition, indicating deficit in sensory motor gating. So this is a summary of the E4 and 3 impact. Heterozygous mice showed shortened synaptic time course, smaller current amplitude, diminished field LTP, with altered dendrite spike, with reduced 2B protein level, with reduced overall 2B mediated excitation and synaptic density. The mice also showed impaired spatial learning memory, deficit of the sensory motor gating, and high anxiety-like behaviors. Dr. Liang lab at Emory helped on the PET scan, and the Gawley lab helped on the spike analysis. Catherine Ross at NDS helped on the immunoblot, and Gordonville helped on the molecular dynamic simulation. The variants presented here from Dr. Limicki's group and from Dr. William Gall from AH. Thank you very much.